Hello, and thank you for joining me. I'd like to explain to you today a few things about a paper that we just recently published in the journal Nature Communications. It's about the structure of an empty MHC class 1 protein, and it's been a really exciting collaboration between 4 research groups that do very different things. The crystallography group of Rob Meijers in Hamburg, the computational biology group of Martin Zacharias in Munich, a mass spectrometry group of Charlotte Uetrecht, also in Hamburg, and then my own group. I'm Sebastian Springer, molecular biologist at Jacobs University in Bremen in Germany. Now, let's get right into it.
Major histocompatibility complex class 1 molecules present high-affinity binding peptides to cytotoxic T cells on the cell surface. These peptides have a low dissociation rate because they fit really well into the tightly defined pockets in the binding groove, where the A pocket binds the amino terminus of the peptide and the F pocket the carboxyl terminal side chain. Understanding these binding mechanisms is necessary to develop peptide vaccines and understand the cytotoxic T cell response to infection and cancer. In order to learn about peptide binding, it would of course be good to know what the class 1 molecule looks like without the peptide, that is, in the empty state. The problem is that no one knew how to make peptide-free class 1 molecules because the binding site is unstable in the absence of the peptide. So peptide-free class 1 molecules have been modeled instead of crystallized. But that does not represent the actual shape before peptide recognition, because the modeled structure is derived from the loaded molecules. This paper now introduces the first ever crystallized empty MHC class 1 molecule. It was made by introducing a disulfide bond between the alpha helices close to the F pocket. The group showed that the disulfide stabilized class 1 molecule shows the same binding affinities and binding interactions to the T cell receptor as the wild-type class 1 molecule. This was shown for the allotype HLA-A2, and it was published a couple of years ago already. But to get the structure, we needed to work with all the collaborators that I mentioned before. First, the wild-type class 1 and the disulfide stabilized class 1 molecule were expressed in bacteria, and a high concentration of dipeptide was used to refold the molecule. The molecule was then purified by size exclusion chromatography. The disulfide stabilized class 1 molecule was then tested on its stability. By thermal denaturation assay at different dipeptide concentrations, the denaturation temperatures were determined. The results show a stable melting point of 62 degrees for the light chain and 1 melting point for the heavy chain that is sensitive to the dipeptide concentration. From this, one can conclude 2 things. First, that the dipeptide glycyl methionine stabilizes the peptide binding site, and second, that the disulfide stabilized class 1 molecule is more stable than the wild-type class 1 molecule if there is no or very little glycyl methionine, which means simply that the disulfide stabilized class 1 molecule does not need the stabilizing dipeptide. It is stable in itself when empty. To demonstrate that the peptide binding site of the disulfide stabilized class 1 molecule really had no dipeptide in it, mass spectrometry was performed after removing any peptides by size exclusion chromatography, presenting the still stable molecule. Finally, it was shown that the binding of a full-length peptide to the disulfide stabilized class 1 molecule is not affected. Therefore, we can trust for the purpose of this paper that the disulfide stabilized class 1 molecule is a peptide-free, stable version of the wild-type class 1 molecule. After the disulfide stabilized class 1 molecule was introduced, the researchers then investigated its structure with dipeptides bound in the A and F pocket of the peptide binding groove. The disulfide stabilized class 1 molecule was folded in the presence of 2 different dipeptides, glycylmethionine and glycylleucine, and then crystallized, resulting in high-resolution crystal structures. The disulfide stabilized class 1 glycylmethionine structure formed in the presence of 1,2-ethane-diol, and the disulfide stabilized class 1 glycylleucine structure was formed in the presence of glycerol. The ethane-diol and the glycerol were components of the buffer in which the proteins were crystallized. The disulfide stabilized class 1 glycylleucine structure showed that one dipeptide is bound in the A pocket and one glycerol molecule from the buffer in the F pocket. The disulfide stabilized class 1 glycylmethionine structure binds two dipeptides, one in the A and one in the F pocket. These structures show that the disulfide stabilized class 1 molecule binds the amino acids in the same manner as the wild-type class 1 molecule binds the residues of longer peptides. The groups then, of course, wanted to crystallize a complete empty version of the disulfide stabilized class 1 molecule. With the empty protein, Rob Meijers obtained a crystal structure of the disulfide stabilized class 1 molecule at a 1.75 angstrom resolution with an empty peptide binding groove, where one ethane-diol is present in the A pocket. The lack of peptide in the A pocket revealed a conformational change in some side chains, expanding the pocket. The hydrogen bond between histidine 70 and tyrosine 99 is released, and an upwards movement of phenylalanine 9 is detected. The new peptide receptive conformation is called the unlocked state. In the second structure, where two ethane-diol molecules are present in the A pocket, phenylalanine 9 appears in the same conformation as in the peptide-bound structure, and histidine 70 and tyrosine 99 engage in weak hydrogen bonding. This state resembles the peptide-bound state called the locked state. The triad phenylalanine 9, histidine 70, and tyrosine 99 accommodates peptide side chains and switches from unlocked to the locked state upon protein binding. Similar conformational changes are presented in the F pocket through the rearrangement of the triad arginine 97, histidine 114, and tyrosine 116. The volume increases from the locked into the unlocked state, resulting in the ability to make space for binding bulky side chains. Lastly, an explicit solvent molecular dynamics simulation was performed to access the stability of the empty disulfide stabilized class 1 molecule. No significant conformational changes were observed. The glycylmethionine dipeptide bound in the F pocket showed greater mobility than the one in the A pocket. The reported fluctuation indicates that there are no tendencies of unfolding even for empty disulfide stabilized class 1 molecules. The umbrella sampling free energy simulations on the crystals gave insight into the free energy barriers between the locked and the unlocked state. The conformational space of the tyrosine 116 side chain dihedral angle was investigated. The gauge conformation corresponds to the F pocket's unlocked state and the trans to the locked state. Both conformations represent a local minimum of free energy, revealing a significant energy barrier between them. This simulation supports the previously stated allosteric model where the triad arginine 97, histidine 114, and tyrosine 116 overcomes the significant energy barrier and switched conformations upon peptide binding from unlocked to the locked state.
So to conclude and summarize the story, our paper has introduced the structure of a peptide-free class 1 molecule, HLA-A0201, in its disulfide stabilized form. And with this structure, we've been able to show that when the peptide binds, the side chains of the amino acids that line the peptide binding groove that those side chains, they undergo some conformational changes, and they do it together. And this set of conformational changes we call the switch between the unlocked state, that's peptide-free, and the locked state, that is peptide-bound. In the A pocket, those residues are phenylalanine 9, histidine 70, tyrosine 99. And in the F pocket, those are arginine 97, histidine 114, and tyrosine 116. They move together. And this is supported by MD simulations that show that there's a significant energy barrier. So it's not just a floppy back and forth, but it's actually a click, click, an energy barrier between those two states. Now, the two other interesting things. We know that the presence of the peptide is required to maintain the overall structure of the class 1 molecule. So that means when there's no peptide, then also the alpha helices or the beta sheet, they might partially unfold. And this, of course, we cannot see in our crystal structure because our crystal structure, our empty protein, is stabilized by the disulfide bond. So that question of the partial denaturation of the secondary structure elements, that remains open. And the other thing is, of course, that there are so many different allotypes. I just recently looked this up. There are 20,000 different allotypes of class 1. So the switch mechanism that we've discovered, that does probably not apply to all of those molecules because they have different amino acids in the binding growth. But we know that it's valid, it operates in the most frequent class 1 allotype in the world, which is HLA-A2. And this just tells us that there is so much more work to do. Thank you very much.